these proteins present. It does not tell how much of the protein is present. When you have talk of targeted, you talk of something that is extremely important in any scientific study, that is accuracy, precision, reproducibility. Also, we have the lower limit of quantification and upper limit. So LLOQ and ULOQ. So these are things that comes up. These are terms that comes up when we talk of targeted proteomics. And as we had already defined, discovery is kind of data centric. So we try to ask the question, what is changing in my samples? So with respect to cancer and non-cancer, or maybe treated versus non-treated, old versus young, what is the difference? Whereas in targeted proteins, I am trying to really target onto a specific protein, sometimes even onto a specific peptide, right? Suppose you're trying to design an assay for a viral peptide. You want to design an assay to detect whether a particular person is having a viral infection, right? One way, always we have the RT-PCR. What if you can detect viral peptides in his blood, or a malarial parasite peptide in his blood? So once the peptide is present, it's more like an ELISA or Western. Only this time you can upscale it. So why do we need to go for a targeted approach? There are 4 reasons. First of all, the dynamic range. Yesterday when we were talking about sample preps, we were talking that how we have to deplete high abundance proteins. Suppose you are working on high abundance proteins, and you're also wanting to study both a highly abundant protein and a low abundant protein in the same assay. With discovery approach, you cannot do, because either you can detect the highly abundant protein, and then if you are detecting the highly abundant protein, you will mask off the low abundant protein. Vice versa. To detect the low abundant protein, you have to deplete the highly abundant protein. But with targeted approach, you can just take your raw sample, get the peptides, and then just by specifying the instrument that, okay, these are the 2 proteins I want to look into, you can do this. So you have solved the problem of dynamic range. Precision, it's very important how much of a protein is present. You can do that. Cost effective, of course, when you don't have to go through a depletion column, you are effectively saving a lot of money just in sample processing. And since it is highly precise, and the reproducibility factor also increases. And this is one of the reasons why proteomics has not strongly impacted in the clinics, right? You might see that there are multiple genomics clinics even in India. And abroad, if you go, you would find people are, just for the sake of it, sequencing their own genome. Especially you have cases like Angelina Jolie, who got to know what will be the predisposition for a cancer, and multiple people are doing it. In first world countries, we have the money to do it. Even in India, there are particular labs that they have started doing this. Whereas if you see proteomics, even abroad, there are very few options. But there are few options. These are the very few options that has started off. One is OVA1-TM. So it is a prediction of cancer risk in women with adnexal mass. You have DCP and FEF13 that gives you a risk assessment for development of hepatocellular carcinoma. You have Roma, you have PCA3, you have Overa. So all these are biomarkers that are FDA approved protein biomarkers that you can look into. And the goal of doing science, especially with the respect of our biology and medicine, especially in the medicine part, is finding more such biomarkers, taking it to the market, make your research more translational. So there are multiple approaches when we talk of targeted. So there is one thing that we know as the selective reaction monitoring. You have multiple reaction monitoring, so SRM and MRM. Also you have parallel reaction monitoring, that is PRM. You have SWOT MS based techniques, that is again a DIA approach. But as I said, multiple instruments have multiple specifications, and your workflow will also be quite influenced by the instrument you're working with. And what do you do? So Skyline is a very famous software tool that we would briefly discuss. we don't have opportunity to go deeper into it, but maybe in some next GYAN course we can discuss Skyline in detail. So SRM is the work on the selected ion chromatograms. PRM works on the extracted ion chromatograms. So does DIA swath, and DDA is for the extracted ion chromatograms of MS1. So we talk of the word tandem MS, MS. So MS1 extracted ion chromatograms are, so what do you do is, you take the DDA, LC, MS primary data, you make a spectral library. What is a spectral library? It primarily is tells that these are the spectra, these are the peptides that could be detected in your sample. And remember, not all peptides will be detected by the instrument. For a given protein, not every peptide, and you know the reasons we discussed yesterday why we cannot detect all the peptides, right? So much of our studies are basically, what is the length that we need to have? Between 7 and? You have exams today, so you should have studied harder. So 35, 7 and 35. So, so you do a selection of target peptides, then you go to the SRM and PRM methods, you set up the method where you just detect only those specific 8 to 10 product ions, right? So now you are cutting down the job for the instrument. The instrument is not looking for the entire peptides, all the ions. You are selectively telling the instrument to look for only those guys with red hats, right? If I put you up in a traffic junction and tell how many people are, or how many cars are passing by, it will be a very difficult job for you. But if I give you a job that, okay, count the number of yellow cars, that immediately the load on your cerebral cortex will significantly come down. Or maybe I just tell you count the number of red hatchbacks or red sedans. Then again, so this is in very dumbed down format what a targeted proteomics approach tells. So we, using the software, tell Skyline, and we do a method optimization, and we select only 3 to 5 ions, right? We select very less targets, and this makes it very precise. The instrument is only looking for that specific signal that should come from the sample. So of course, this is the workflow for a targeted. So you know, you do proteolysis, you do peptide separation, you do electrospray ionization. Then what you do is, this is a typical exposition of a, do you have the laser pointer, volunteers? Anurag, the laser pointer. So what you see, Q1 is the mass filter. Now see, here I have put a mass of 0.7 m by z. There are multiple balls, right? You have red, orange, green, blue, purple. But what I am selecting is only the balls that are green. That has a charge by mass ratio of 0.7. Mass by charge ratio of 0.7. What I do in the next quadrupole, it's a collision cell. So here we do some HCDs, high energy dissociations, but these peptide fragments are broken down further and it generates Y ions and B ions. Now this Q3 is again a mass filter. This is a triple quadrupole, right? Q, Q, Q. So this is a quadrupole, this is a quadrupole, this is a quadrupole. So in this final quadrupole, since I have selected only 0.7 m by z, only these specific dots are detected. So it lowers the computational load, and these collision cells and the mass filters, so we have 2 mass filters and 1 collision cell, so they specifically help you into a very targeted approach. And you see, of all these precursor ions that we got, we are only looking for this trace fragment ion. So this is why we call it the selected reaction monitoring, right? So that's the reason. So if you again go back to the terms, we have SRMs, PRM. I'll come to PRM very soon. Now what if during the SRM run, what happens is MS continuously acquires spectra at the expected m by z, right? and precursor ions are selected from Q1, fragmented in Q2, and target specific fragment ions are selected in Q3 part. So this is the basic workflow. I think it is clear to you. We will discuss further when we have more time. So again, selected reaction monitoring. So again, the same things. So you have mass filters that will select for you, right? So you are standing at a traffic junction, right, to make things easy for you. So suppose you are 3 friends, right? So you are standing at a traffic junction. Your first friend will allow only allow all the cars to come in. The next friend, for better color detection, is standing with a jet spray of water. So all cars that pass through the Q2, they are sprayed. Now you are only standing at Q3. This is another mass filter. So now you can very specifically detect that how many red cars passed through. There is no issues. Maybe there's enough light for you and you can give a good detection. This is a very analogy that you can think of that we do in targeted proteomics experiment. Now, if you don't want to use a mass filter like triple quad, if you want to use an orbit trap mass analyzer, what you have to do is you can use something known as the parallel reaction monitoring. So this is again almost a similar approach, but here our mass analyzer is an orbit trap. It's not a triple quadrupole. So you have a precursor ion. There you have a mass filter. It's like a quadrupole. Then you have a collision cell. You are breaking down these same ions, and in the orbit trap mass filter you are detecting the specific ions that you need to look into. Is the video on? YouTube links are on? YouTube. YouTube पे WhatsApp पे. So PRM is also referred to HR, high resolution SRM, or HR stands for high resolution SRM or MRM, because it involves the use of high resolution mass spec, especially orbit trap and TOF analysis. So the same technology, the fundamental remains the same. When you upgrade the technology, you can use an orbit trap or a TOF analyzer, and it exploits the trapping ability of instrument for better resolution and increased sensitivity. So the idea of a targeted experiment is that you have a very predefined hypothesis that I want to detect a specific particular parasitic antigen in the blood of my patient, or a specific protein that is known to increase the predisposition for cancer in the patient plasma. So that makes it very easy for the instrument. And since you already have, once you start understanding Skyline, although this is not part of the syllabi, all of you can look up on Skyline, and there are very good YouTube tutorials by Brendan. So you can look up SR Skyline, and they have a very good, their YouTube channel is filled with tutorials. And if you're only into targeted experimentation, because most of us don't actually go till targeted. So what PRM does, it reduces the assay development time, it eliminates most interferences and enables confident confirmation of peptide identity, because you are doing multiple reaction, you are doing parallelly SRM, so it's more like high resolution SRM. So there it reduces the assay development time. So PRM developments, so SRM, it takes about maybe 15-20 days in a very well established lab. It will take over a month to develop, because you will go through the entire samples, you have to create the lower limit and upper limit, you have to quantitate with labeled peptides, all these heavy labeled peptides. So you have to do that. PRM significantly, because it has a higher resolution and sensitivity, can reduce your workload. So what usually people do, this is by Brendan. So what people usually do is you take you can use servers like Panorama, and again, this is an ecosystem, Skyline, Panorama, where you generate your own data, you upload to Panorama, and they, based on your instrumentation, will suggest, okay, these are the peptides these instruments can detect properly. So since we are not going deeper into it, I'm just giving you a brief. So what is the ecosystem for, and these are all open source softwares. If you want to learn, you can learn it from your home. They have enough tutorials, and they have also some data sets that you can use to train yourself. So basically, I will just tell you now how to perform a targeted proteomics experiment. So there are a few methods by which you can make it. So first is the RT libraries. What is RT? Retention time. So since we're even a triple quadrupole will work on the principle of chromatography, and we'll have something known as the retention time. Then you have the spectral libraries, the spectrum that it has been generated for each of your sample. Then you can use methods and previous measurements, and you do a lot of targeted method refinement. So this is where you spend most of the time, because in your first discovery data, so you usually start with the discovery data. The spectral library is generated from a discovery set data that you have done this entire sample, and in this entire sample for one specific protein you could detect, suppose, 75 peptides, right? And out of these 75 peptides on a triple quadrupole, all 75 might not be detected, right? So you will take the spectral library of 75 peptides, you will use the retention time libraries, and then you will do a targeted method refinement. Out of these 75, you might come down to only 3 and 4 peptides. But these peptides are very robust. Every time you inject the sample, it will detect at a very low limit of quantity. You can also quantitate how much of your target protein is present by creating this simple standard curve that you do in protein quantification, right? So you can do quantification, you can do very sensitive detection. And how do you do it? So you have protein targets, and that you can generate from the peptide atlas, the UniProt server, from KEGG, and you create, for these targets, you have peptides. As we discussed, for one protein, you have multiple peptides. These peptides are what? Made out of trypsin proteolysis. Then you create the transitions. Transitions means how each peptide is broken down further in the dissociation cell, what kind of Y ions and B ions they produce. Then you do the LODs, the LOQ, the calibration curve. Then you create a linear range. So this is more of a quantitative profiling. Then you try to assure the specificity, accuracy, and precision. And then you also design the assay range and robustness. How much of the sample you need, how strong your assay is, like is it working on all sample types, is it working on only specific sample types, right? So suppose you're talking of detecting viral peptides. Now when the pandemic came, we came to know that even lions and tigers and monkeys are getting infected, even minks, all mammals kind of became reservoirs for the virus. Suppose you're trying to look at a pest, something that is zoonotic in origin, and you have a reservoir host and you have your patient. Now, can you detect in both? Can you take a bat blood and detect the peptide, or is it working only with human blood? So all these kinds of assays, because when you develop an assay, it has to be robust. It should work each time, every time, and it should work with precision. So this is primarily from this paper. You can look at it. So when you design a targeted proteomics experiment, you have, so this protein, you see protein 1, protein 2, these proteins are not generating peptides from every fragment, right? So you have specific peptides. You select these specific transitions. So look at the yellow colors. So these are the transitions from the mass spectra that you are selecting. And then you optimize what is the fragment, what is the retention time, and what kind of data can you generate for each of these specific peptides. So one protein can generate multiple peptides, and from out of those multiple peptides, all you need to do for a robust data is about 3 to 5 peptides. If you can analyze and make your assay very robust, you can take it to the clinics. So this is just, I will go through this in very briefly. So you start with a protein sequence search. I think all of you know this is a database, is UniProt. Then suppose you search for P53, which is a cancer specific tumor antigen. Then you upload it on Skyline. Now Skyline, based on the protein sequence and based on the enzyme that you will add on the database, from the database, you know the protein primary sequence is there, right? And Skyline, you can computationally detect that, okay, these are the peptides that can be formed. Next, they will create these kind of precursor ions. So you have Y2, Y3, Y4, Y5, Y6, Y10, till these are the ions. We'll discuss about these ions sometime very soon. So you see, this is the fragmentation pattern. So these are the Y ions, these are the B ions. It's just when you cut the NH2 side is the B ion. So for this specific fragment of peptide, if the dissociation is here, the B1 will carry F and the Y11 will carry the rest, right? So this is the basic schema of Y ion and B ion formation. This happens in the collision cell, right? So high energy dissociation, so this dissociation just breaks up the peptides. So you see, out of all these, you have multiple factors like what is the M by Z at Q1, what is the M by Z at Q3. Q1 is the first quadrupole, the mass filter, Q3 is where we are detecting it. Then you have the fragments, so you can detect Y5, Y7, Y8, Y10, and these are the dwell times. So dwell time is in that trap. How much time is that ion particularly spending? So it's 250 milliseconds. So it's a very short span of time, and this entire acquisition, so accumulation and acquisition time is 250 milliseconds for each of these sequence. So if you add it up, that becomes the cycle time. Again, you have to number of refinements that you have to make sure. What is the abundance? We discussed, right? Is your peptide or protein highly abundant or is it low abundant? What is the measurability? Can you measure the peptide? I mean, not all peptides will fly. Is it prone to degradation? The digestion that you are using, the trypsin, are you using trypsin? Sometimes you can't use trypsin for specific reasons. So that will, scheduling, optimization. So there are multiple ways by which you can do a targeted method. Refinement, it's a long process. Discovery is quite easy. You go till trypsin digestion, you clean up the peptides, and then you allow mass spec to do all the stream. When you want to do a targeted approach, these targeted approach are fairly difficult. The reason they are difficult is because now you have to refine and you have to specify that this is the peptide, but since they are also difficult, they are very precise because you have spent a lot of time optimizing. So these are the outputs of Skyline. So if you can see, these are the specific ions. Is it visible to all of you? So this is the first one is Y7, then Y6, then Y5, Y4, Y3, Y5. So this is the specific peptide, and these are the retention times that could be, these are the specific ions, and these are the charge on the peptide. So this is the result output, and this is what a selected reaction monitoring looks like. So for one specific peptide, you get very sharp peaks like that. And if you're getting such peaks, that means you have created a very robust assay. So the robustness of your assay is shown by these peaks. And again, one more thing that you need to think of it, because since you already have your computational data that is coming from the Skyline database, right? Skyline will predict what could be its retention time, what would be the data you are achieving. Then there is an experimental data, which of course will be different from what we have already achieved. You might have, those who are into experiments would realize that whatever is written in the books is fairly different from what you get. So that gives us the dot p value So dot product value, so dot P or dot product value, a perfect is 1. It's a perfect correlation. Whatever Skyline predicted, you got. You have a value of 1. But the range is from 0 to 1, and just like correlation, anything less than 0.8 is fishy, anything less than 0.5 is extremely doubtful. So, but again, a lot will depend on you as an experimenter, that what you set up as the baseline. If you're trying to detect something, maybe in animal biology, you can go for some lower limits. But suppose your decision means life and death for a patient. Your decision means a patient might get an ICU bed or might not get an ICU bed. Right? You have taken up a prognostic tool and you are telling that this specific person might get into severe dengue in the next 24 hours. That could mean a life and death. So there you have to really have a very high dot P value, so that we'll try to achieve 1. 1 is a perfect score. Nobody achieves that. But you see, these are 0.99. So these are actually, you can go till 0.99. This is 0.82. So these are specific dot P value for this target protein RAS2. This is again the MS spectra that is showing the same. So I think I will end here, because the rest part is not very much necessary for you, but I would love to show. This is something that we did at IIT Bombay along with, this is Professor Sanjeeva's slides. So we wanted to see that if we can detect COVID, the SARS-CoV-2 viral peptide in nasal swabs, right? Because they're already mass spec. So you might ask, there were already PCR based studies, but PCR later on we understood gives us a lot of false positive results, right? Proteins are much more robust in terms of detection. So what we did was, we took some positive samples and we tried to develop an SRM assay, and here we could detect the SARS-CoV-2 nucleoprotein and SARS-CoV-2 glycoprotein in the samples. So of course, because there is a lot of work to do as far as infrastructure development in hospitals are concerned, and mass specs are still very, mass specs are extremely very, I would say a costly instrument to handle and costly instrument to maintain. So there is some issue with translation to clinics, but actually if you go to labs, tests are ready. Maybe we need a more cheaper intervention, more cheaper or more scale development. Sometimes, like real-time PCR was a real novelty back in 2005, 2006. Even till 2018 it was a novelty, or until the COVID pandemic happened, and suddenly there was so much production of real-time PCR instruments that now real-time PCR instruments are really cheap. So I think I will stop here, and we can, where is the webcasting team? You can pause the recording. So if you have few questions, we can take it. Maybe I will take 1